Alya, very nice talk. So the first part of the talk, where you were looking at the translocations of nerves through that basal membrane, do you have any idea how important that movement below the basal membrane is relative to actually wrapping the nerve with the Schwann cells? So do you have any mutants where the Schwann cells fail to make basal membrane, but they still wrap the nerve so the nerve doesn't fall apart, the mutants or doesn't move? Or some of those other animals you had where the Schwann cells made basal membrane but stayed in the epithelium, but those not move as much as the ones that didn't have Schwann cells?
Right.
Or those nerves didn't move as much?
Yeah, so in the cases where Schwann cells are present, we do see less defasciculation. Is that what you're asking? Like individual axons being frayed away from the bundle?
Right, even though they stay up in the, like the nerves where they had Schwann cells, but they didn't go down like they were supposed to. Those nerves still get pulled as much?
They do a little bit, but not as much. So you can, I've always gotten into arguments about my lab about this, but I swear there's a little tiny dip here in the nerve. So even though there are Schwann cells present here, and these nerves will actually probably move, I can see by EM also sometimes that the nerve is actually not localized where it's supposed to be. It's often sometimes located ventrally when there isn't a complete rescue of Schwann cells along the lateral line nerve. Whenever the Schwann cells make it totally to the end of the tail, you never see defects in the kind of DV axis. But whenever there's just a partial clone of Schwann cells, we did sometimes see that the nerve was still being pulled despite the presence of Schwann cells.
Okay, so there's some additional component of actually having to localize it below that basal membrane.
Yeah, so it might be a timing thing. So like maybe these guys didn't get localized early enough to avoid being tugged, or maybe because the axons down here are still in the epidermis and they're being tugged, it's actually having effects on the nerve up here. Yes.
I really enjoyed your talk. So from the first part of your talk, I think in the transplant experiments, if I understood correctly, there is always the formation of a new basal membrane, but the old one is not always degraded. So what are your thoughts about how is the old one degraded if it's not by the Schwann cells? And also in wild-type cases, is there a crosstalk between the two to ensure that actually there is no double basal membrane?
Those are questions that I would love to know the answer to. So what my best guess, I guess, would be that, well, we thought it might be matrix metalloproteinases because they're known to have roles in degrading extracellular matrix, and we know that they're expressed in Schwann cells. And I actually did an experiment where I inhibited MMPs, but we didn't actually see any defects. So it still could be MMPs, those would still be my best guess, and we just didn't hit the right ones. In terms of, so that was the first part about how it might be degrading it, and that would be my guess that actually the Schwann cells are secreting something that's actively degrading it. In terms of crosstalk between like degrading the old one and making the new one, they're happening at the same time. So I'm going to guess that there has to be something important about Schwann cell polarization, that it has to know which side is out and which side is in. And it has to know that either to do it itself or induce the formation of a new basal membrane only on the outside and to degrade only on the inside. But beyond that, I have no idea. Are you really going to ask me a question?
Yes, I really like the idea of this being, of the movement through the basement membrane being a common motif, and I was wondering what your thoughts are on finding other systems where that might be true, and like showing that it happens this way.
Yeah, so we did, I did look at the taste buds in superfish, because believe it or not, fish have taste buds. And they look remarkably like neuromass. The problem was I couldn't actually tell, there were axons everywhere, so I couldn't actually tell whether in the absence of Schwann cells we lost axons on one side or the other, because we don't know enough about taste buds yet to really know which axons are coming from where. But I wouldn't be surprised if maybe you need Schwann cells in other cases for, like in the dermis, for an axon to be able to push through that basement membrane, because in your, in the innervation of your epidermis and also in your tongue, the nerves actually grow out below the epidermis, below that epidermal base membrane in the dermis, and then they have to make a 90-degree turn and poke up. And there are Schwann cells associated with those nerves, so it would be cool to see if maybe you need Schwann cells in order to actually bust through the base membrane and make it to the nerve, because the Schwann cells will get all the way up to the base membrane and then stop, and the nerve goes through.
So what you're saying is that during development, in that case, it goes the opposite.